the hematopoietic system at homeostasis and how we can study the evolution of cell clone during steady state hematopoiesis. But before we get to that, it's not working. Okay, how do I advance slides? Oh, there. Yeah, good. So I want to say happy birthday to John. So one of the highlights of having been at UC Irvine for 20 years was to have John as a colleague and as a friend, and it's been really amazing, and I'm really happy to be here to celebrate this birthday. I've also gone through some photo albums. Here we are. I'm showing off our reproductive fitness. I still have hair there, which is kind of interesting. That was a long time ago. Now these people are all grown up. It's hard to believe. I still have that sweater. I just noticed. Okay, so moving on. Maybe somebody can turn the lights down because I don't think you can see very well the slides. So this is the system that I'm going to be talking about today, the hematopoietic system. The hematopoietic system is maintained by hematopoietic stem cells. This also, there you go. They're on the top of the hierarchy. The stem cells undergo complex differentiation pathways to give rise to myeloid progenitor cells, lymphoid progenitor cells. These then differentiate further to eventually give rise to the cells that comprise the blood. And so this is one of the better understood systems where we kind of have a lot of understanding of the stem cells and the differentiation pathway and their dynamics. Also, tumors evolve and emerge in those compartments. Different tumors of the blood exist, and they arise in different stages of this differentiation hierarchy. Some of them develop in the blood stem cells, in the hematopoietic stem cells, such as CML, chronic myeloid leukemia. Others are thought to arise more in the progenitor compartments, and yet others more in sort of more differentiated compartments. And so to understand this whole thing better, it's important to kind of understand how mutants, aberrant cells emerge and evolve in a hematopoietic system that is in a healthy state, because that's the first step in carcinogenesis. And so a very, very interesting phenomenon in this respect is something that is called clonal hematopoiesis of indeterminate potential, also CHIP, in the blood. And that basically refers to the emergence of mutant cells in a non-malignant blood system. And interestingly, some of these mutations are characteristics of cancer, so they are found in blood cancers. But in this case, in the CHIP phenomenon, you see these mutants pop up, but there is no cancer. The specific mutations are TET2 or DNMT3A. These are involved in methylation, and they can be followed in the clinic. And so as people age, higher and higher fraction of the population carries these mutations. And so what is the consequence of these mutations? The most immediate consequence of the mutations are that there are some metabolic disorders, cardiovascular complications. And this arises primarily out of the fact that these mutant cell clones that sort of emerge in the blood system cause inflammation, and that has all sorts of knock-on effects. It's not good for the health. But a small fraction as well of these patients that carry these mutations go on to develop hematopoietic malignancy. And it's kind of unclear what the meaning of these CHIP mutants is, what the risk of further carcinogenic development is, but it is definitely there. But this is something that is subject to investigation. And so let's look at some data of how this looks like. This is a very kind of confusing dynamics. There are lots of things going on. So on the y-axis, you see this VAF. This is variant allele frequency. It basically tells you the frequency of these mutations in the blood over time. And I want to concentrate on this picture. It's a little bit clearer. And you can see that there are various patterns that you can see. These mutants pop up. Sometimes they grow and they keep growing. Okay. In other cases, they pop up, they're there for a while, and they decline and go away. And yet other cases, they pop up and remain relatively steady over long periods of time. So you have some complex evolutionary dynamics that are going on in the blood, and people are investigating this from various angles, but it is not really understood what is going on here. So this is just sort of the summary of these things. So you see this initial increase, and they kind of steady out, and then they can either increase further, stay steady, or actually decline and disappear. So we wanted to look into this with the help of mathematical models. And we wanted to ask a much simpler question. So these are complicated long-term dynamics already, but we wanted to understand what are the conditions for a mutant to actually pop up in the hematopoietic system. Assuming that these mutants have some advantage over the wild type, under what conditions do we expect them to pop up, and in which compartment of the hierarchy are they expected to pop up? So we started to look at basic models of stem cell dynamics. And this is just a very, very crude representation of some of the model assumptions that we have. So what we assume here is that there is basically symmetric cell division going on, and a stem cell can give rise to two daughter stem cells, or it can give rise to two differentiated cells. And this happens with probabilities, and these probabilities have to balance out. There's obviously asymmetric cell division that can be going on, but there's increasing evidence that this is sort of the probabilistic scheme is something that's quite prevalent in mammalian systems. So this is the basic principles we have. This is the basic, very primitive model of stem cell dynamics, where the most important processes are taken into account. You've got stem cells that divide. When they divide, there's a certain probability P that they self-renew, give rise to more stem cells. And with a probability 1 minus P, they give rise to two differentiated cells, and differentiated cells can die. Yes?
I'm sorry to interrupt.
No worries.
Yeah, it's just it's new area for me, and I certainly realize you use a lot of probability theory. Natasha also used some concepts, but here what you said, it's actually it has a special name in this area of stochastic processes. Specifically, it's called binary branching.
Okay.
Yeah, so you
Yes.
create another name for it.
Well, I haven't created a name for it. Just say
Yeah,
that.
yeah, but it's actually, it
Yeah,
has formal name
yeah, yeah.
already.
Okay. Yeah.
Just comment, which I hope would be useful.
Right, sure.
Uh-huh.
Sounds good. Thank you.
Sorry to interrupt.
Yeah, no worries. So this is the basic scheme that we have. So what I want you to remember from this is that there's a division, and the division can be self-renewal, the division can be differentiating. And there's been quite a bit of work done on these sort of models. John has been very much involved in this, other folks at UC Irvine too, and in the hematopoietic system, the groups of Anna Masiniak and Thomas Stiel have been all looking at similar things, where in order to get an equilibrium, you need feedback processes that are happening in the system. So if you have these probabilistic divisions, and in order to have a biologically realistic stable equilibrium, you need feedback where when there are more differentiated cells, for example, you feed back on the self-renewal probability, and that keeps the population at a constant homeostatic levels. There are various feedback mechanisms that can occur. They have been documented experimentally and also theoretically modeled in various ways. Some of them can maintain homeostasis, whereas others do not contribute to homeostasis. So in the following, what I'm going to do is concentrate on feedback factors that contribute to homeostasis. So here we are. This is the model that we have been using, that we have been applying to the blood. And so the hematopoietic system is a very complex system. I showed you this picture with many, many cell types. We are restricting our analysis to those cell types for which we have data. And I'm going to come to the data in a minute. We have data from mice that gives us a lot of insights into the kinetics of the system. So we start off with the long-term hematopoietic stem cells, LT-HSC. These are at the top of the hierarchy. Then these differentiate to give rise to the short-term stem cells. They're a little bit further down. Then you get the multipotent progenitors. And then you get common myeloid and common lymphoid progenitors. And we'll assume that these are the last parts in the differentiation hierarchy because we don't have any more data. This can be either death, this arrow out here, or differentiation to further downstream compartments. So it's basically the same kind of model that I showed you before, but more detail. We have more compartments in here. Also, again, we have the division rates and we have the self-renewal probabilities, and we have to assume that there's some sort of feedback, and you can do it generally. And we did a general mathematical analysis where we assume that any of these cell types can produce feedback factors that will inhibit the self-renewal capacity of any of these cells. We can assume a very simple, specific for demonstration purposes. We assume a specific feedback. We assume that each compartment secretes factors that feed back on their own self-renewal capacity. And so this results in an equilibrium in a homeostatic setting as we observe it in the blood. We have these feedbacks expressed by these functions here. You have a basic self-renewal probability in the absence of feedback, which we typically call C, and we have a strength of feedback, which we call H. So that's basically the system we're considering. And so now parameterizing the model. The model has to be parameterized in order to give biologically important insights. So we did that. I'm going to come to that one. Then we examined some basic cellular dynamics, which I'm also going to explain. And then we'll move on to the evolutionary part of the talk. So how do we get insights into the kinetics of this process? There's some very interesting mouse experiments that came out relatively recently from Thomas Höfer's lab in Heidelberg. And they have it's called label propagation experiment. So they label the long-term stem cells in the mice. And it's a label that you can switch on when you want to. And when you do that, you can follow the progression of the label from the long-term stem cells to the various compartments that you have there. So you see that increasing, and you can have a time series of the label in those different compartments. And that's highly valuable for parameterizing mathematical models. So you can make a mathematical description of the label propagation data that we have. You can fit that to the data. And by fitting that to the data, you get parameter values of this model that I showed you. And the parameters are shown here, the best fitting parameters. And they kind of make sense. You can see that the division rate of the long-term stem cells is very low. It's like one in 100. They divide once in 100 days. The division rates of the downstream compartments becomes faster. Obviously, the self-renewal probability of the long-term stem cells is 0.5 because it has to be like this. If it wasn't 0.5 at equilibrium, at homeostasis, it would either increase or decrease. So that's 0.5. The self-renewal probability of the downstream compartments, short-term stem cells, and MPPs is less than 0.5, and we're going to come to that as well. If you look at that, the conclusion is that they can't maintain themselves in the absence of the long-term stem cells because these differentiate more than they self-renew. Okay, so they got a bunch of parameters, and we proceeded with that. Feedback, you can't really measure the feedback, and we don't really know what all the feedback factors are, but we can, given that we have all the other parameter values, we can have combinations of the self-renewal probability in the absence of feedback and the strength of feedback that are consistent with the number of cells in the different compartments that's been measured in the mice. And that's what we did to kind of guesstimate feedback parameters. So I want to come to the interpretation of those data. So as I mentioned before, the long-term stem cells have to have a self-renewal probability of 0.5 in order to maintain the whole system. Again, if the self-renewal probability was more than 0.5, then they self-renew more than they differentiate. So you get an expansion. If it's less than 0.5, then they will differentiate more than they self-renew, and they decline. So it has to be 0.5. So that's clear. Then what I want to concentrate on is the measured self-renewal probabilities of the short-term stem cells and the MPPs, and these are less than 0.5. And this makes sense. If you look at this, the common notion is that the long-term stem cells maintain the system. So if you cut those out, the interpretation is that those won't be able to maintain the blood system because their self-renewal probability is less than 0.5. So the whole system would just go extinct if you remove that. So we looked at that in a bit more detail, and there's some interesting things that can happen. So we have this parameterized model, we have the system at equilibrium, and now we can delete cells in the computer simulation in silico, right? So here is the equilibrium, these are the cell populations, and here we deplete the long-term stem cells. This is the self-renewal probabilities of the different compartments. So these are long-term stem cells, short-term stem cells, and I believe the MPPs. And so let's see here what happens when you delete the long-term stem cells. Contrary to what the intuition would tell you is that the system does not actually go extinct in the simulation when you deplete the long-term stem cells. So here they adjust a little bit, but they go to a new equilibrium. And remember, in the previous slide, these are the self-renewal probabilities that are measured at equilibrium. They're less than 0.5, so you would think that they actually go extinct, but they don't. And what happens is you deplete the long-term stem cells, then the self-renewal probability of the short-term stem cells starts to dynamically rise and become 0.5, and it acts as a de facto stem cell and maintains the system. Let's go further and deplete also the short-term stem cells in this simulation. Now, again, it doesn't go extinct, and you can see that now the MPPs start to increase their self-renewal probability, and it converges to 0.5. And so in each case, if you deplete the stem cells, the downstream progenitor cells seem to be able to maintain the system in this simulation. And I want to point out that we don't have replication limits in here. So if you introduce replication limits, which we've also done, this is going to be a more temporary state, but it can nevertheless maintain it for a while. So why is that? So this is just a summary, what's going on. These are the measurements. So you've got the long-term stem cells, short-term stem cells, and MPP. Now you get rid of the long-term stem cells. These start adjusting, the downstream compartments start adjusting their self-renewal capacity such that they persist and maintain the system. So why is this? It has to do with the feedback dynamics that we assume in these models. So typically, if nothing is depleted, you have the long-term stem cells and the short-term stem cells and the MPP. Let's look at the MPP compartment. There are two things that are going on. The MPP cells themselves replicate, and these cells make feedback signals that reduce the self-renewal probability of the MPPs. But you also have things moving in from the long-term and short-term stem cells. That's an additional influx of cells. They also produce feedback signals that reduce the probability of self-renewal. So we've got both of these forces reducing the probability of self-renewal, so the P is less than 0.5. Now we get rid of this by depleting the long-term stem cells. Then you get rid of this component that was producing feedback signals, so you have less inhibition of the self-renewal probability, and so the whole thing shifts up to 0.5, and it maintains the system. So that's the explanation for that. And this behavior of the model is consistent with data. There's quite a bit of evidence out there that if you deplete long-term stem cells, this does not really mean the end of the hematopoietic system. The MPPs and short-term stem cells can, to some degree, at least temporarily, reconstitute a system and maintain a hematopoietic system. This kind of could give an explanation why. Okay, so that was an interesting analysis of the sort of dynamics that is possible under these assumptions and interpretation of the measured parameter values. So now we want to turn to mutant evolution. So under what condition do these chip mutants evolve? So we take a model that is kind of exactly the same as before. So this is the model that I showed you before, ODE model, going down the differentiation hierarchy. And here this is the wild type now, and here we have a mutant that has exactly the same processes happening, but they differ in parameters. In particular, we assume that they differ in their self-renewal probability. In this model, a higher self-renewal probability, a basic self-renewal probability, means an advantageous mutant. So we assume that this mutant is advantageous. They compete with each other through shared feedback signals, and parameter S is the selective advantage that the mutant has. So we only look at advantageous mutants, and we will look at different degree of advantages and what they do. So this is the system, wild type and mutants. And now we're going to place the mutant into any of these compartments and ask what is the condition for this mutant to evolve and emerge. Okay, you can place them into various compartments. You can place them in the long-term stem cells. This is a compartment where they are least likely to emerge because, as I showed you before, these cells divide very rarely, and so the probability of actually generating a mutant in here is very small. We can put them into the downstream compartments where they replicate faster. There's a higher probability for that. And so what is the fate of these mutants if you place it into the individual compartments? So here is putting the mutant in the long-term stem cell compartment, which is kind of unlikely, but if you do place them there, as long as these cells have any advantage at all, no matter how minuscule, the mutant cells will rise and will eventually go to very large values and take over. Of course, here this takes a very long time, longer than the lifespan of the mouse, because these cells divide very rarely. But I wanted to, the takeaway is that any advantage will lead to invasion here. Now you place, again, a mutant. We assume the 2% advantage, same advantage here, 2% advantage. We put them into the short-term stem cells. And we see that, oh, actually in the progenitors here, in the MPPs, it doesn't matter. What we see here is that these mutants go extinct. They don't grow. So this advantage that allowed growth in the long-term stem cell does not allow growth in the progenitors. However, if you give these mutants a massive advantage, there's 25% in this case, they will grow, and they will grow in a reasonable amount of time and take over. So you need kind of a monster mutation in this simulation in order for it to take off in these downstream compartments. So this indicates that there's a barrier to mutant invasion in these downstream compartments. And this is shown here, and there's sort of a trade-off that's going on. We can calculate that the mutant invasion barrier analytically, and based on the numbers, the parameters that we have, we could put numbers to them. So in the place where it's least likely to generate a mutant, you don't have a barrier. Any advantage will lead to growth. In the compartments where it's likely to generate a mutant, these mutants are very unlikely to take off. They cannot take off because of the invasion barrier. They need a massive advantage in order to grow. Now, what is a biologically realistic advantage? It's up to debate, but a 1% advantage in biology is considered quite a big mutant advantage. So this indicates that there's an intrinsic resilience in the hematopoietic system against mutant invasion. Here, it's just very unlikely to generate them. They can grow, but it's not going to matter because they grow so slowly. Here, they're likely to be generated, but they can't invade. So you are in a situation where the hematopoietic system seems to be pretty robust against mutant invasion. So why is that? Why is that invasion barrier? What is the reason for this? Again, it has to do with the influx from one compartment to the other. Here is the progenitor cells. Let's consider the progenitor cells. There's two components to the fitness of a mutant. One component is the reproduction within the progenitor cell compartment. And here the mutant cells have an advantage because we assume they're advantageous in the model. Okay, so they do have this growth advantage because they have a higher self-renewal capacity. However, the second component of fitness is the influx from the upstream cells to the downstream cells. And if the mutant arises downstream here, it does not have that influx. But the wild type has an influx from here to here. And so that renders these mutants somewhat disadvantageous as a consequence because they don't have this influx happening. And that is the reason for these invasion barriers that we see. Okay, so I want to finish up the talk by asking, can we use the model to generate some hypotheses based on experiments that will allow the mutants to eventually grow? So we have this identified this intrinsic resilience to mutant invasion. So under what conditions can these mutants grow? And we were looking at two things. The first thing is aging. During aging, the influx of stem cells from the long-term stem cell compartment towards the progenitors gets reduced. That's one thing. And the second thing that we looked at is what is called evolutionary niche construction, where the mutant cells actually can create an environment for themselves that is more and more advantageous for themselves. So let's look at what this is. So this is very simple schematic term. In young individuals, there's lots of differentiation from the long-term stem cells towards the more differentiated cells. But if you age, this input gets reduced. And as I mentioned, this input from long-term stem cells towards the downstream compartment is a major reason for these invasion barriers. So that invasion barrier can go down with age. And the other thing is that this is something that's experimentally documented as well, is a very interesting thing. So you've got the wild type stem cell lineage, the mutant stem cell lineage, and these mutants, when they differentiate, you get to the end product, these produce inflammatory mediators and they cause inflammation. This inflammation and these inflammatory mediators, interestingly, negatively hit the wild type stem cells, and they reduce the self-renewal capacity of these stem cells. However, they do nothing pretty much to their own self-renewal capacity. So these inflammatory mediators put the wild type cell population at a disadvantage and gives the mutant population an advantage. This is called niche construction, evolutionary niche construction, because initially, if there are few mutants, that's not going to matter, but the more they are, the more they make the environment such that the environment is beneficial for the mutants themselves. And so we revisit these simulations. So we have this 2% advantage, and we're looking at the short-term stem cell population now. As I mentioned before, 2% advantage. The mutants here that you see, they don't grow, they go extinct. Okay. Now we put the aging thingies in. So this influx from long-term stem cells to downstream compartment, we just assume that over time it declines in some way, simply due to aging, which is experimentally documented. And you can see now all of a sudden these cell populations, the mutant, don't go extinct anymore, they start to grow. However, they grow very slowly. Okay, so this can't explain the rise of these stems, and that's all under the measured parameters. Okay, so this can't explain the rise of the mutants to significant levels within the lifespan of the mouse. However, if we include this evolutionary niche construction, then they do grow in a reasonable amount of time. And that is initially of kind of the same growth, but the more mutants there are, the more favorable the environment becomes for the mutant, and it kind of is a positive feedback, it takes off, and it will allow the cells to reach significant levels. And so now we have a set of predictions about the conditions that are needed in order for the mutants to invade. And the next step is that those will be tested experimentally. And that's it. I don't want to keep you from lunch much longer. I just want to acknowledge current and former group members. Also, importantly, a high school student has contributed significantly to this work. And from a hematology point of view, with the input of Angela Fleischmann at UC Irvine and Natalia at UC San Diego. Thank you very much.